Hey guys, welcome back to another video. Today we're going to be talking about nucleic acids. So let's get started. So the first thing that you need to know about nucleic acids is that they are polymers. And if you don't know what a polymer is, it's basically just a molecule that's made up of repeating and identical subunits called monomers. So if a brick building was considered to be a polymer, its monomer units would be the bricks that make up the building. Now, if you still don't understand what a polymer is, I have an entire video dedicated to explaining polymers and how they're formed. So go check that out before continuing on with this video. Anyways, moving on. The monomer unit of a nucleic acid is called a nucleotide. And this is what we're going to be discussing over the next couple of minutes. So this is what a nucleotide looks like. It's made up of 3 components. The first is called a pentose sugar. A pentose sugar is also known as a 5-carbon sugar because it's composed of 5 carbons. So there's your first carbon, your second carbon, third, fourth, and fifth carbons. Now, bonded to the first carbon in the nucleotide is something called a nitrogenous base. There are 5 different nitrogenous bases. We have adenine, thymine, guanine, cytosine, and uracil. Apart from the 5 different bases, the only thing you need to know about nitrogenous bases is that there are 2 different kinds of nitrogenous bases. The first category are purines, and the second is called pyrimidines. Purines are the bigger nitrogenous bases. They're made up of 2 rings connected to each other. Pyrimidines, on the other hand, are the smaller nitrogenous bases, and they're only composed of 1 ring. Examples of purines are adenine and guanine, whereas cytosine, thymine, and uracil are pyrimidines. Moving on, the third and last component of a nucleotide is the phosphate group, which looks like this. It has a phosphorus, 3 oxygens, and a hydroxyl. So now we're going to be talking about how 2 separate nucleotides combine to form a single nucleic acid. This happens through a process called dehydration synthesis. Now, I already have a lesson discussing dehydration synthesis reactions, but for the sake of this video, I will give you a brief definition. Dehydration synthesis is when 2 monomers are combined by removing a water molecule. So you remove H2O. In this case, the hydroxyl bonded to the 3rd carbon of the first nucleotide, and the hydroxyl in the phosphate group of the second nucleotide combine to form a water molecule and an extra oxygen. Now the water molecule leaves, and the oxygen is left behind to bind the 2 nucleotides together. So the molecule ends up looking like this. So all of that's gone, and you have a single oxygen binding the 2 together. And this happens again and again, and more nucleotides are added to the nucleic acid, which ends up making the molecule look like this. So a very common nucleic acid that we all know about is DNA, or deoxyribonucleic acid. Now DNA is double-stranded, and it takes the shape of a double helix, which looks like this. So the two strands that make up a DNA molecule run anti-parallel to each other. What this means is that the two strands are parallel to each other, but they run in opposite directions. So a molecule of DNA doesn't look like the molecule on the left here, it looks like this. So notice how on the left strand of the DNA molecule, the third carbon, or the fifth carbon, is at the top, and the third carbon is at the bottom. But on the strand on the right, the fifth carbon is at the bottom and the third carbon is at the top. So on my double helix over here, I would say one strand runs from its 5' to 3', and the other runs from its 3' to 5'. And prime is just a fancy way of saying carbon. I'm saying it runs from its fifth to third carbon, or third to fifth carbon. But let's go back to the nitrogenous bases for a second. Notice how the nitrogenous bases pair up in the molecule of DNA? Well, there are actually specific pairings. So let me just erase this over here. So adenine pairs up with thymine, and guanine pairs up with cytosine. So if I were to have this nitrogenous base be adenine, then the other nitrogenous base would have to be thymine. If this one over here was cytosine, then the other would have to be guanine. So far, we've only discussed DNA, but there's also another very important nucleic acid that you need to know about, and that is called RNA, or ribonucleic acid. So in this last part of the video, we're going to be discussing the differences between DNA and RNA. So the first difference between DNA and RNA is their functions. DNA stores genetic information. It holds genetic material. And basically what this means is that DNA controls everything about you, from the color of your hair, to your height, to even the way you behave in certain situations. RNA, on the other hand, is involved in protein production. It's basically a very specific set of instructions on how to make different kinds of proteins. Now, the second difference between DNA and RNA are the nitrogenous bases that they consist of. So DNA consists of adenine, thymine, guanine, and cytosine. RNA consists of adenine, uracil, guanine, and cytosine. So RNA swaps out thymine for uracil. A third difference between DNA and RNA is that DNA is double-stranded, whereas RNA is single-stranded. So instead of taking the shape of a double helix like DNA, RNA would take the shape of a single helix. Now, the final difference between DNA and RNA that you need to know about is their sugars are different. DNA has a sugar called deoxyribose in it, whereas RNA, its nucleotide consists of a sugar called ribose. Ribose and deoxyribose are essentially identical, except for the fact that deoxyribose has one less oxygen in it. So say I have a ribose sugar here. I'm not going to draw the entire thing, but just know that you have a hydroxyl over here. Deoxyribose doesn't have the oxygen in that hydroxyl, which is why it's actually called deoxyribose, as in less oxygen ribose. And that's all you need to know for this video. If this video was helpful, please like it and hit that subscribe button down below. If you have any questions, please comment them and I'll do my best to answer, or even if you have suggestions for new topics you want me to explain in future videos. As usual, I have linked a checklist for review in the description box down below. Good luck studying and bye!